Okay, so as I was saying before, that proteins can be large, complicated molecules, just because for the first part, they have 20 different amino acids that can be arranged in different ways. That gives you a lot of potential combinations. But then once they are formed into their final chain, then they twist into this helix-like structure. A helix is kind of like DNA. And then these structures also form into sheets, and the sheets fold and twist and bind all together, and you get a unique shape at the end that's determined by the chemical properties of each amino acid. This gives it a very complicated structure, very complicated 3D structure. Okay. Unfortunately, we didn't have enough time to do this during our lab, but it's possible to denature proteins. To denature a protein means to destroy it. So if you play around with the pH, things become too acidic or too basic, or there's too much salt content, or the temperature gets really too high, then the bonds that hold the protein together start to break apart, and it loses its shape, and then it's no longer able to function properly. Okay, so something that can, if the amino acid sequence is wrong or it has a deformed shape, it can have very kind of fatal consequences. So one of these that we looked at was sickle cell anemia. Over here you see a fairly normal red blood cell, and then this is one of the sickle cells, right? It loses its ability to carry oxygen properly around the body. But as we did talk about in class, that this does seem to have some sort of survival advantage, sickle cell anemia, in that it does provide protection against malaria. Okay, the last big thing that we talked about that will be on your test are enzymes, and they are specific kinds of proteins that carry out chemical work in the body. So enzymes are proteins. They're what are known as catalysts. And a catalyst in chemistry is anything that can speed up the rate of a chemical reaction. And the interesting thing is it not only speeds up the rate of the chemical reaction, but is not particularly involved in it in the sense that it's not used up in the chemical reaction. It's still there afterwards, and yes, it can be used again. So enzymes speed up chemical reactions. A lot of these biological reactions that they speed up would take place anyways. They just happen far too slowly. So this is what we did in class, right? We looked at amylase, right, which is found in your saliva. It can break down sugars, starch, into component sugars. There's the enzyme protease, which breaks down proteins into constituent amino acids. So how does it work? So we just need to understand this in a very basic sense. So we have our enzyme. Our enzyme has a spot on it called an active site. This is where a chemical can bond to the enzyme. The chemical that bonds to the enzyme that undergoes a chemical reaction or chemical change is called the substrate. Substrate bonds to a specific enzyme because of the chemical shape and nature of the active site. So it says the shape of the active site only fits the shape of the substrate. And this is what is called a lock and a key mechanism. Just the way a key fits a certain lock, a certain active site will only fit a certain substrate. So we've got the enzyme binding to the substrate, we've got a chemical reaction that's occurring, which is always about breaking chemical bonds or forming new ones, and then the products are released. Again, very much in a kind of lock and key mechanism. Lock and key mechanism. The nice thing about this is, unless we're dealing with extreme temperatures or pH changes, enzymes are not permanently changed or used up as reactants. They just provide a site where chemical reactions can occur, and they go faster. Okay. We kind of talked about this before briefly at the beginning, the idea of activation energy, and that's an idea that we'll definitely look at more if you decide to take an AP chemistry course. But how catalysts or enzymes work is they lower the amount of energy that's needed for the chemical reaction to proceed. So most chemical reactions follow this kind of pathway, in that the products have a lower energy state than the reactants do. Most chemical reactions all chemical reactions need a certain amount of energy to get them started. This kind of chemical reaction is an exothermic reaction in that it's going to release energy, probably in the form of heat. But you can see without the enzyme, it requires a lot more energy to get started than without the enzyme. So if you want to speed up enzymes even more, speed up chemical reactions even more, what can you do? Well, of course, you can add more of the enzyme that you want. You have more enzymes. That means you can do more chemical work. Or what you can do is you can heat up the enzyme as well. You can warm it up. So this graph over here shows you enzyme activity. And for this one, around 40 degrees Celsius, it seems to have a kind of maximum ability to act. Anything above 40 degrees Celsius, the enzyme's activity falls off. Why? Because if it's warmed up too much, it begins to denature, which means that the chemical bonds within the protein, remember that enzymes are proteins, they'll begin to break down. Okay? Likewise, most of them have an optimum pH that they work at as well. If the pH is far too high or far too low, the shape of the active site can change, which will affect the activity of the enzyme. Okay, so just to recap, enzymes have 4 special features in common. They are catalysts, meaning that they speed up chemical reactions. They're not used up after reactions the same way catalysts are. They can be used for forward and reverse directions of the chemical reaction, and it's very highly selective about its substrate, meaning only certain substrates will bond to the active site of certain enzymes. Okay, you should also review our lab that we did. Okay, this kind of finishes up Unit 1. So for Unit 1, what do you need to look at? You have a lot to do for your test. You have things about atoms to look at. You have things about the characteristics of life to look at. You have the particle theory and classification of matter to look at. And then you have all of these macromolecules that we looked at as well. All of this should put us in good shape for our next unit when we start actually talking about the cell. Okay, good luck.